Good morning, everyone. My name's Andrea O'Connor, and I'll be giving you a lecture this morning, so it's great to see you all today. I'm from the School of Engineering, so I'm going to be telling you about some of the work that we do that relates, I think, to what you've been hearing about in the last week or so around stem cells and tissue engineering. This is an area that I've worked in for quite a few years now, and I collaborate very closely with a lot of medical researchers, biologists and clinicians. So it's a very fun area to work in. I've only got an hour, so I'll just give you a snapshot of some of what we do in this space. And particularly I've been asked to focus on biomaterials and some of the directions in biomaterials. Before I kick off, just for my interest, how many of you are doing the bioengineering systems major? Fantastic. A few future engineers in the audience. Maybe I can persuade some more of you. So, yes, I'm in biomedical engineering. We've got a new department of biomedical engineering just formed this year, but we've been teaching in this space for a long time, and you've got my email there if you want to get in touch with me. So, as I say, I work in the tissue engineering field, and what I want to talk to you about today is particularly the roles and significance of biomaterials in tissue engineering. So, this is an important and reasonably complex issue. I want you to appreciate the design criteria for some of the processes that we use to select biomaterials that we might use in tissue engineering, how we fabricate and tailor those to be suitable. I also want you to understand the key limitations and challenges of using biomaterials in tissue engineering. So there have been many mistakes made with biomaterials, and if you pick the wrong material, things can go awry. They won't work well. So there's some complexity there. We need to understand a lot about the features of these materials to get the most out of them. If anyone has questions, feel free to put your hand up as I go along. So just to set the scene, I'm sure you're familiar with tissue engineering as a concept. These two gentlemen, Bob Langer and Joseph Vacanti, were credited with kicking off the field. Around the early 1990s, there was discussion about how engineering and physical sciences could contribute to this problem of patients losing or having failing organs or tissues. So they came up with this definition in their science paper in 1993 that said that the loss or failure of an organ or tissue is one of the most frequent, devastating and costly problems in human healthcare. And this new field, tissue engineering, applies the principles of biology and engineering to the development of functional substitutes for damaged tissue. Now that word functional is very important. We don't necessarily need to make an exact replica of the tissue that's missing. If it's an organ like perhaps the pancreas, it doesn't need to look like the pancreas looked originally. It needs to function in the way that the patient needs it to function. So understanding that function is an important part of this equation. So tissue engineering as a field aims to generate new functional tissue to repair or replace tissues that are missing, and that might be due to disease, particularly cancer, it might be due to genetic defects or trauma like vehicle accidents. So there's many reasons why patients need replacement tissue and situations where their body can't heal that tissue on its own. If any of you have ever had a severe break in a bone, you'd be familiar that it doesn't necessarily heal all that well, and sometimes you need surgery and maybe even plates and screws or something else to help that bone heal. So tissue engineering aims to overcome those kind of challenges. It's got great promise. So people have been very excited about this opportunity that we might be able to get over tissue shortages. We don't have enough donor tissue, donor organs to treat all the patients who need treatment. We might be able to get better results than what we can get with current medical devices or other surgical options. We can customise implants. So particularly for patients who have perhaps a major trauma, we might be able to use medical imaging data to get an image of the defect where their tissue is missing. I work with a company that does this on people's skulls. So if you're in a major car accident, they might need to remove part of your skull to allow the brain to swell. When they come back to put something in that gap, we can use medical imaging to get an exact image of what that gap looks like and customise an implant. So that will be a better result for the patient than if we just took something that was roughly right off the shelf. And there's the opportunity to create new treatments where we don't have any good treatments. So spinal injury is a classic example of that. Can we use tissue engineering to overcome severe spinal injury? So there's great promise in this field, and there's been huge excitement, and the media every now and then gets excited and reports that massive developments are happening. But there's been limited outcomes, if we're realistic. The number of tissue-engineered therapies in the clinic is still reasonably small. So one of the things I want to do today is ask why. Why aren't we seeing all this amazing promise fulfilled in the clinic? Well, there are hurdles, of course, like any new therapy, and tissue engineering is complex. So we have technical challenges, and I'll talk some more about some of those today. But there are also significant commercial challenges about how you get funding, how you create new companies, how you overcome the risk of this field. And there are regulatory hurdles, because the regulator, like the Therapeutic Goods Administration or the FDA, are still developing their rules as the technology develops. It's difficult to predict what can be approved for clinical use, and this is a challenge as well. So just bear in mind that it's not all technical challenge. There are some business and commercial and regulatory challenges as well. And I think you've already heard something about the ethical challenges from Megan Munsey around stem cell therapies as well. So let's think about the technical aspects. Our tissues we can think of as comprising three main components. There are cells, there's some kind of matrix, the extracellular matrix that sits around those cells is very important, and there's a blood supply in almost all of our tissues. Cartilage is an exception, but for our other tissues the blood supply is very important. So traditionally, tissue engineers simplistically thought, well, if we could get some kind of scaffold, a sponge, and make that in the shape that we needed, add cells, maybe add growth factors or other molecules that would help those cells to grow. We could culture that in the laboratory, the cells would stick on the surface, they would grow, and voila, we'd have a new piece of tissue that we could implant into the patient. This has been tried a number of times. Generally it doesn't work. So there's lots of reasons why this can fail. It's difficult to get your cells to do exactly what you want. It's difficult to get the material to behave exactly the way you want, and things are somewhat unpredictable. So this seemed like a great idea, but it's overly simplistic. And one of the key things we're missing here is that blood supply. So I'm sure you're aware that your cells in general need nutrients and they need oxygen supply. So in this situation, there's no allowance for providing that oxygen supply to the cells once they're implanted in the body. So that's a big reason for failure of tissue engineering attempts early on. So the strategies that we take depend on the type of tissue that we're trying to regenerate. So we need to think more specifically about what's the situation, what's the tissue, and we can develop in vitro constructs if the tissue is avascular, like cartilage, or if it's very small or very thin, so we can say that it's approximately two-dimensional. Then mass transfer of molecules can carry oxygen and nutrients to the cells. But at human scale, in 3-dimension, we need a blood supply, and we need that early so that our cells don't die waiting for it. So typically cells will suffer from hypoxia within hours or maybe a day or so. They can't wait a week for new blood vessels to form. So one strategy that we've been involved with to overcome this is actually growing the tissue in the patient's body. Rather than trying to grow it in the lab and transplant it to the patient, we use what we call in vivo bioreactors to allow the construct to develop inside the patient's body. If you like, we're using the patient's body as a bioreactor. And that helps us to overcome some of those challenges that you would have trying to engineer everything outside the body. And this diagram gives you a schematic of one of the experiments that we've done in a small animal where we have a blood vessel. We work closely with microsurgeons who can do very clever things with connecting blood vessels here. So this is connected to the host. We have a chamber, which is just a plastic housing that we put inside the animal, and over time, small blood capillaries sprout from that main vessel, and tissue starts to grow. So the green here is tissue developing, the red are the blood capillaries sprouting out from that original loop. So by doing this, we started off with no cells in the chamber. Cells have come via the blood supply or through the gap at the edge of the bioreactor, and after about 6 weeks, they can fill this chamber with tissue, and the tissue is vascularized. So this is a strategy that we have investigated, and I'll show you later in the class where we've taken that. So we've got some key challenges in our tissue engineering strategies. We need a blood supply for three-dimensional vascularized tissues. We need suitable biomimetic matrix materials, so we need to pick good biomaterials that will help our cells to grow and do what we want. We want to deliver the right kinds of signals, physical and chemical, biochemical signals to our cells, and we also want to avoid infection. So with any implanted material in the body, the risk of infection is a significant complicating factor. So we're interested in how biomaterials might help us to avoid or limit that problem. So all of this leads us to think that we need tailored design of systems for tissue engineering, taking account of which kind of tissue we're trying to engineer. So what's the role and the significance of biomaterials in all this? Well, we can use biomaterials to provide scaffolds, like sponges for the cells to attach to. They provide surfaces and microenvironments that will help the cells to grow and do the things that we want, like lay down extracellular matrix. So they provide a microenvironment for the cells. Ideally, they mimic the extracellular matrix that the tissue has in its native state. So as an engineer, I want to understand that ECM very well so that I can try to make materials that will replicate properties from that. So they provide space and that biochemical environment to allow new tissue to grow. So they're providing that opportunity for tissue to grow, providing signals to help that to happen. And note that the mechanical properties are very important as well as the chemical and biochemical properties. So over the last 10 years or so, it's become increasingly clear through some exciting biology research that stem cells respond to their mechanical environment, and mechanical forces make a big difference. So we can exploit this in our biomaterials design. So what are our design criteria? How are we going to pick biomaterials? There's hundreds of options out there. So what are our criteria and the processes that we'll use to select, fabricate and tailor these materials? Well, there are lots of criteria we might think about. And for good design, we're likely to have to compromise. We probably can't have everything that we would like. When I talk to the surgeons I collaborate with, they often give me a list of properties that just seems impossible to achieve. So we have a discussion about, well, is this the most important thing? And what if I could only get half of that process? Would that be okay? So we need to compromise and optimise. And to do that, we need to understand the tissue development process. So the designs will depend on our target tissue. If you're trying to grow skin, it's going to be very different to trying to grow bone. Different mechanics, different cellular environment. Something to be aware of and careful of is you often see people say, oh, this material is biocompatible. So people will use that word very widely, often without good justification. So it's not a universal property. I could say that a Band-Aid that I put on my skin is biocompatible for that purpose. If I'm not allergic to it, it may work fine, it may not cause any adverse reaction. But if I implanted that inside my body, I expect that it would cause an adverse reaction. So how you're applying that material makes a difference to whether you can call it biocompatible. So the application makes a difference, and you need to be careful of claims that materials are biocompatible. Check how that's been tested. How did they decide that that was the case? And there are lots of novel materials being developed all the time. So there's a lot of applications for biomaterials, not only in tissue engineering, also in drug delivery, medical devices and implants. So people are developing things like smart polymers that respond to their environment, they change in some way. Bioactive glasses, these are materials that will bond very well with bone, so they're good for healing in bones. Porous hydrogels, macroporous 3D hydrogels, I'll show you some of those. Composite materials with multiple functionality, hierarchically porous materials with different sized pores, and of course 3D printed materials are becoming more significant in this field. So lots of developments. We need a strategy of how we're going to pick what we want to use. So some criteria for biomaterials specific to tissue engineering. We want our materials to be biocompatible for the application that we plan to use them in. So we need to think about what tests should we do to see if that is a valid claim. They need to have mechanical properties that are suitable for our target tissue and the implantation site. So skin obviously has a softer property than bone. So for bone you would have a stiffer material. And then if you consider bone, there are different loads on your bones in your body. So in your leg bones you need a very high strength as you run and jump. In the skull the forces are less. So the site of implantation makes a difference. The biodegradable nature of the material. In tissue engineering, ultimately we would like synthetic or added materials to disappear. So eventually they have degraded away and we're left with a piece of tissue that's well integrated with the host tissue and we can't even tell where it is. So it naturally fits into the body. So we'd like the materials to biodegrade, and so we're worried then about how long does that take, how rapid is it, the strength of the material as it degrades. Does it suddenly lose its strength or is it a gradual process? And what byproducts will it release? So a number of biomaterials can work very well for a period of time and then they might suddenly release, for example, acidic components. So there have been a number of cases where everything seems to be going well and then after 6 weeks or 12 weeks, acid is released due to the degradation of the material and the growing tissue dies. So we need to be careful about that aspect. We want suitable in vivo responses. So how will the body respond to this material? And one of the major challenges we have is dealing with inflammation. Our body is very good at trying to get rid of foreign materials. And in tissue engineering, we're typically trying to implant a foreign material and we don't want that to be subject to the foreign body response, FBR. So that can be a problem for us as well. How do we design a material that won't cause a big foreign body response? We want them to be able to be fabricated into structures. So it's all very well to have a great material, but if I can't make it into the object that I need, that's a problem. It should be cost effective, available so that I can purchase supply of the material, and able to get regulatory approval. So if you start with a material that's never been used clinically, it's going to take quite a long time for you to get regulatory approval to be allowed to use that in patients. If you start with something that has been used maybe for a different application, the pathway to regulatory approval is a little bit easier. And that's part of the reason why older biomaterials tend to get reused, even though perhaps they're not perfect in their properties. They need to be able to be sterilised. Another big challenge in tissue engineering, because we're often using materials that are similar to the extracellular matrix, and it can easily be damaged by sterilisation techniques. So we're working on research to improve the sterilisation techniques. Ideally, our materials should be stable and have a reasonable shelf life. This comes back to the commercial aspect. If you have to make your materials just before they're used, commercially that becomes very difficult. But if you can have a stock of these materials available in a hospital, then they can be more readily used and the cost becomes more feasible. And we'd like these materials to actually promote the desired response from our cells and tissues. So that might be proliferation of cells, differentiation, certain gene expression. So the materials may be able to help direct the behaviour of the cells. So that's quite a list of things to try and achieve. There's probably not one off-the-shelf material that would be perfect in all of those. And then you can probably think of some more criteria as well. So this is where we may have to compromise. Another challenge we have is we may not actually know in detail how all of the materials will behave on all of these criteria. So we've got some limitations and challenges. Potential problems include the foreign body reaction. As I said, this is the body trying to get rid of foreign materials. We can have acid release, so byproducts from degradation of materials. Maybe toxicity, some materials will actually kill our cells. The supply, the cost, and the reproducibility of our material. Is it always the same every time I make it? Is every batch the same? And then this lack of knowledge of our design criteria. We may not know exactly how rapidly a material biodegrades in the body or what its strength will be over time. So this complicates our design. And then lack of predictability in vivo. So can we predict how the body will respond to our material? How much inflammation will we get? What kind of cellular responses will we see? These things are difficult to manage, and this is part of the reason why there's still a significant amount of animal testing in the field of tissue engineering. So there's research ongoing to try and replace animal testing as much as possible, but these challenges make it very difficult to predict from in vitro tests exactly what will happen in vivo. And even from small animal tests to larger animal and larger animal to people is still difficult. So ongoing research in those areas. So I'd like now to show you some examples of biomaterials that we and others have developed for tissue engineering, to give you an idea of the kinds of processes and properties that we can obtain. So here are some tailored porous biomaterials. On the right-hand side are scaffolds. These are scanning electron microscope images of the insides of some polymer scaffolds. They're all made of the same polymer, one of the polymers that we use for degradable sutures. So this is polylactic glycolic acid, and they're made by a process of freezing, and in the freezing process we get crystals of solvent that form, and the reason these are all different is the way that we did the freezing. So just by changing the process, we can change the internal structure of the materials. So we can think then about how would that help cells. Is there enough room for cells? Is there direction, such as in this one, that might guide cells that would normally grow in a directed way, like myoblasts or maybe nerve cells might like a structure like this. So we can control something about the internal architecture. Here's another one down the bottom left showing you an emulsion templated material. So a different kind of internal structure. And these gels here are different compositions of a particular gel, and you can see just from looking at them that they're different stiffnesses, and that's shown in this graph here as well. The compressive modulus is a way that we characterise how stiff the materials are, and this is polyacrylamide gels. You can see very soft at part A through to stiffer versions, and the elastic properties of these are comparable to the elastic properties of our soft tissues. So we can tune the mechanics of materials, we can tune their porosity, and these other properties as well. So the architecture of the pores, the stiffness, the degradation properties, other chemical and physical properties. We can choose different chemicals, different materials, and we can work with polymers. Most of the molecules we're interested in our body are polymers, and we also have ceramics, which are useful particularly for bones, because our bones contain hydroxyapatite, so we can use ceramic materials to mimic that. Hydrogels are very important. These are water-swollen networks of polymers. So these polyacrylamide gels here are examples of hydrogels. They contain a lot of water. So that's like our extracellular matrix. And we can make composites. So there's a wide range of options here. Let's look then at one example of hydrogel production. This is to make macroporous hydrogels. So those gels on the previous slide didn't have holes in them. But if you want your cells and blood vessels to penetrate into the gel, it would be a good idea to provide a network of pores or holes to allow a pathway. So this is one way that we can make a porous gel. Here we're starting with a solution. This process is called cryogelation. So we call this the cryogel solution. We have here the polymer, HA is hyaluronic acid, and we have a crosslinker, EDC for short. And we take this solution and we freeze it. We cool it down so that the water in this solution will freeze. So we form crystals, and those crystals will grow until they bump into each other, and we'll get some chemical cross-linking because of this EDC molecule. So we end up with a gel that's chemically cross-linked, and it has these ice crystals in it. When we warm it up, the ice crystals melt, and we're left with a porous gel. So we have then a concentration of the polymer that occurs when we freeze, and that drives the cross-linking process. And because we've only used water to make the pores, we don't have toxic components to remove. So we can get very high porosity, about 90% pores, so only 10% or so polymer, and very well interconnected pores. So there are pathways then for cells to move through. And we can tune the properties, and they're low toxicity because we're not using toxic components. So this picture shows you a very soft gel through to a stiffer gel. We can control the properties very nicely. And this is a nice engineering process because we can scale it up. So we could do this at a very small scale, but we could also imagine doing this at a manufacturing scale. So economically, it should be viable. So here's some of the mechanical properties. This is a measure of the mechanical stiffness, the storage modulus here, as a function of the polymer concentration. And as we add more polymer, we get stiffer gels. And you can see here that the stiffness of these gels roughly corresponds to these tissues, fat, brain, kidney and heart tissue, across this range of moduli. So we can tune the mechanics of the gel that we make to at least roughly match the tissue that we want to grow. So that's a nice tunable and controllable system. The next example is additive manufacturing or 3D printing. There are many developments going on all the time in this field. This is a printer that we recently purchased in my lab where we can print polymers, we can print ceramic pastes, and we can also print hydrogels with or without cells. So this is a nice system because it's very flexible, and it basically just squirts out the material that you have in the cartridges into a dish, and you can make that a bath if you want. That will provide an environment where the material will solidify. And we can keep cells alive through that process and then put them into an incubator to develop and grow a piece of tissue. The next example then is antimicrobial materials. I mentioned infection as a significant challenge. So how do we make biomaterials that will help us to overcome that challenge? Well, one way we can do that is to use alternatives to antibiotics. We know that antibiotic resistance is becoming an increasing problem. So in this work, we've used nanoparticles of inorganic materials that are also antimicrobial. So we have what we call a sol gel process. This is a chemical process that allows us to produce nanoparticles, and it can be done at the same time as producing a scaffold. So we can do the scaffold production with a polymer, for example, and at the same time produce nanoparticles which will be dispersed throughout the scaffold. So these white dots on this electron micrograph show you the nanoparticles spread out throughout a biodegradable scaffold. So why do we want to do that? Well, we can add components like silver. Silver is known to be antimicrobial, and see that the number of bacteria present, this is colony forming units, count here as a function of different materials. The colony forming unit count can decrease in the presence of a very small amount of these nanoparticles within a biomaterial. We can use these to make coatings. So, for example, we've investigated coating these kinds of nanoparticles onto orthopaedic fixation devices. So we don't want infections to occur when you have a major surgery like an orthopaedic repair. So if we coat an antimicrobial component that is long-lasting onto those materials, we can significantly reduce the risk of infection and particularly biofilm formation on the surfaces. Now, silver is not probably the best material to use. It's very effective against bacteria, but it's not very friendly to mammalian cells. So it also is somewhat toxic to mammalian cells. And in fact, we're working on other inorganic nanoparticles which are less toxic to the mammalian cells, and some formulations that even give us selectivity about which bacteria will be attacked. So that's an area of ongoing development. But I want to move now to this foreign body reaction. So we've got a few examples of very nice biomaterial systems. We can tailor those to particular applications. But we need to be very careful about the foreign body reaction. This will undermine and stop our tissue engineering efforts if we're not careful. So it's a normal reaction of a higher organism, like a human, to an implanted synthetic material. And this really limits biomaterial implant performance and particularly tissue engineering regeneration. So, in case you're not familiar with it, this is a schematic diagram of how the foreign body reaction would occur when we implant something into the body. So, if we implant that material, very quickly, within a matter of minutes, proteins absorb onto the surface. So, the surface that we may have designed changes very quickly. Proteins will absorb onto that surface unless we've designed the chemistry to stop that. And then cells will start to interact with that material. And they will sense that this material is not a normal part of the body. Even though it's coated in proteins that come from that person, those proteins are likely to have denatured. So even though they are the patient's proteins, if they are denatured, the immune system will recognise that that doesn't belong there. So neutrophils and macrophages in particular respond within a day or so. And then those cells will find that they can't consume and dispose of that material. It's too large. So they'll fuse to form giant cells, and they release significant amounts of cytokines, signalling molecules that send out messages to other cells. And in response to that, lots of fibroblasts tend to accumulate at the site, and they synthesize extracellular matrix components, in particular collagen. So over a couple of weeks, lots of collagen is produced in this environment. We've got lots of fibroblasts, and this leads to the material being encapsulated in a bag made of collagen. It's walled off from the rest of the body, and there's very few cells inside that area. So what we wanted to have happen was cell infiltration, blood vessel formation, and good integration with the surrounding tissue. But in fact, what we have is our material isolated from the rest of the body, fibrous tissue around it, and it's not integrated. So this is the exact opposite of what we want to have happen in tissue engineering, and a number of attempts to use biomaterials have led to this problem. So we want to think about how could we design better materials that would stop this. I mentioned PLGA earlier. This is a material with a long history of clinical use. So there are many good aspects of this polymer, but it causes this foreign body reaction. So this is a histology slice of a scaffold, a PLGA scaffold that's been in a body for a couple of weeks, and the white area is the polymer, the PLGA. The blue shows you where there are cells and fibrous tissue, and the interesting thing here is this one very large foreign body giant cell. It has multiple nuclei because it formed from a number of macrophages, and this gives us a strong indication that there's a foreign body reaction to this material. So the body is trying hard to get rid of this material, and this will lead to adverse outcomes with our tissue engineering. So we thought about, could we still use this material but change its surface so the macrophages wouldn't respond in that way? And one way you can do that is to coat things on the surface. So we wanted to maintain the strength and the processing properties of PLGA, but once we'd formed a scaffold, we wanted to coat the surface, effectively to hide it from those cells. So here's our polymer surface down the bottom, and we can go through some chemical steps. Again, we're designing processes that lend themselves to scale up. So they lend themselves to potential for manufacture of these materials at a larger scale. We can change the surface chemistry to introduce hydroxyl and amine groups. This gives us some anchor points. We can then add other molecules. So we chose hyaluronic acid. As we saw in the hydrogel example, it's a nice polymer, a high molecular weight polymer that is present in our bodies, and it has a lot of negative charges on it. So it's polyanionic, and it will be attracted to the amine groups on this surface just by electrostatics. So we've got a coating then of polymer on the surface, but one layer is not enough. So after that, we put on a layer of polycationic material, and we chose chitosan. This is not a polymer that comes from the human body, but it can be broken down in the human body. So positively charged molecules, and we can build this up layer by layer. And if we do a number of layers, we can really effectively cover that polymer surface. So now the cells will sense hyaluronic acid when they see this surface, not PLGA. We can cross-link that so it doesn't wash away in the body. So chemical cross-linking with EDC again. And then we have a surface that is more like the extracellular matrix. We put samples of this into the body. Here's our scaffold structure, and here are two histology images after implantation in a rat for 2 weeks. This is the bare material, so no coating on the surface. And what we see is this big white area is the polymer with very few cells inside, and we're getting some capsule formation around the outside. But when we look inside, in this area where there are cells around the boundaries, we see cells that stain with a marker ED1. And that's a marker for macrophage cells. So this tells us, when there's lots of this brown staining, that in fact there's a significant inflammation process going on here, and we're likely to head to a foreign body reaction. So this is showing us that PLGA on its own is not giving us a desirable response. But when we coat the PLGA scaffolds with the layers of polyelectrolytes, the response changes a lot. So now these are zoomed in images of the polymer scaffold, these worm kind of structures, and you can see actually some of the layer by layer coating as this hairy material on that surface. And you can see there are some cells in there that stain brown, but there's a lot less, and they're tending to be rounded. They can't stick on the surface. So they're in there, but they're not causing that escalation to the foreign body reaction. So we regard this as providing a kind of blank slate. We've turned down that foreign body reaction, and now we can add particular molecules to bind the cells that we're interested in, or maybe release growth factors or other components. Another strategy that's recently been developed is to actually then provide ligands for cell binding. And one of the PhD students in my group has recently published this paper where she was able to cluster these ligands together. So we're making actual islands of ligands, and this can direct the cell interaction. So some sophisticated functionalisation of the surfaces then to provide some areas that are non-adhesive and some areas that cells will attach to very well. And by providing clusters of ligands, we can get integrin clustering in the cells, and that improves how the cells interact with the surface. So here are some cells on that surface showing that they can spread and attach very well. And this graph just shows you the cell density on different materials. So the different coloured bars here show you different numbers of ligands in those clusters. So going from yellow to green to blue, we're increasing the number of ligands in an island, and the cells attach more readily. So we get more cells attaching to that surface. And this is particularly attractive for blood contacting devices where we want endothelial cells to attach. So lots of developments in how we engineer these surfaces. Another thing we can do is deliver bioactive molecules. So we can add in some biological signalling molecules in there. And in this example, we're doing the same layering of polyelectrolytes on PLGA, but we've added in a sandwich of heparin and fibroblast growth factor. We sandwich these together because the heparin protects the fibroblast growth factor from denaturing. And we can do this inside porous structures. So we favour these kind of wet chemical techniques because they allow us to do it in larger structures and to scale it up. This is how we make the porous spheres. So we're using an inkjetting technique, a customised inkjetting technique where we squirt out droplets of a polymer solution that are very even in size. We have an inkjet nozzle here, and that's over here. You can see it's controlled by a piezoelectric transducer, and we freeze those droplets. So we squirt them down towards a bath of liquid nitrogen. Those droplets freeze, and it's just like the freezing process we saw earlier. The solvent and the polymer separate, and we end up with a porous bead, and that's what they look like once we remove the solvent. So we can make nice porous beads of a uniform size and structure, and we can functionalise the inside of those. So this is just some release data of what happens to the molecules when we're releasing them from those kinds of carriers. And we can tune things like the number of polyelectrolyte layers and how tightly we cross-link them, and it has different effects on different molecules. So here's a small peptide. Alpha MSH is a small anti-inflammatory peptide, and it is strongly affected by the number of layers of polyelectrolyte we put on the surface, but not much by the cross-linking concentration. The opposite is true for fibroblast growth factor. It's a larger protein molecule, and it is not very sensitive to the number of layers, but is affected strongly by how tightly we cross-link that mesh of layers on the particle. So we have different behaviour from different molecules, and that can be understood based particularly on the size of the molecule. So we've got a schematic diagram here that gives you an idea about that. Increasing the number of layers doesn't have as much of an effect on the... increasing the number of layers has a significant effect on the alpha MSH, that's the small molecule here, so less of that will come out, but it doesn't affect the FGF very much, whereas increasing the cross-linking concentration has a stronger effect on the FGF. So we're using size as a way to distinguish and control the release of these molecules. In vivo, we see that these molecules are still bioactive, so it's important with any material development to check if you've actually damaged the biological properties. And we found that the brown staining, again indicating high levels of inflammation, is present with PLGA on its own or with the alpha MSH provided just free with the PLGA. But when we layer that up and protect it so it's released more gradually, we see a decrease in the brown staining, and that's what's plotted here in the graph underneath. When we have the layered coating over the alpha MSH, the amount of brown staining and inflammation has decreased. So we can start to regulate the in vivo responses to biomaterials. And a further example I just want to show you before I finish up is how we're scaling up tissue engineering. So at the beginning I showed you that schematic diagram of the small bioreactor that we used in a rat model. Here is an example where we've placed this into a larger animal model to test if we could grow larger volumes of tissue. So these are in vivo bioreactors. We do some engineering design to come up with the right mechanics and also think about the biomaterial choice for these chambers. We can 3D print these chambers to be exactly the size and shape that we need and place them into the patient with a blood vessel and a small amount of tissue. This is work that's aiming towards breast reconstruction for women who've had mastectomies, and so we want to grow fat as a soft, space-filling tissue. So we have a blood vessel with a little bit of fat provided, and after about 12 weeks, we see that that chamber has filled up with tissue. So this is demonstrating this in vivo bioreactor concept, and when we look inside that piece of tissue, we can see that a large proportion of it is fat. In this example, not all of it was fat. There is some fibrous tissue around the outside, but we're able to grow a larger piece of tissue. This is about 70 millilitres, so it's getting a little bit larger than normal lab scale. And in fact, in more recent times, we've taken this through to a clinical trial where we actually did this type of approach on some women who had had breast cancer, and in the best case, we grew about 200 millilitres of new tissue. and the patient was very happy with that. So we demonstrated the proof of concept that this can work in people. It's not consistent, so there's a lot more research, a lot more development to be done, but the proof of concept is there at least. So let's finish up then. Just to conclude, I hope you've realised that we need to tailor our tissue engineering strategies to targeted tissues. We can do lots of interesting nano and micro engineering to enhance the outcomes from the surface to the material design, the bulk properties, and we need to link our technical developments closely to clinical and commercial realities so that we understand the whole context and actually get these therapies through to patients. Now, if you're interested in this field, I've given you a few references. Number 4 is new since I sent in the lecture notes, so you might like to note this is just a recent review for 2007 in Tissue Engineering B as well. This is just an annual review that comes out. And if you're really keen on tissue engineering, come and do my master's level subject. Thank you. I'm happy to take any questions. Does anyone have questions? Okay, no worries. I'm happy to talk to you afterwards as well. Thanks.